evening, everyone. It's such an honor to welcome you all to the Irvine Barclay Theater. Tonight, we're celebrating 30 years of public lectures here at the Irvine Barclay Theater. That's 66 lectures that have reached over 50,000 community members right here in these seats. But tonight is not just about celebrating 3 decades of impactful lectures. It's about honoring a legacy of scientific discovery that continues to shape the way that we understand the brain, and of course, about celebrating the vibrant community that we have built together. This lecture series began with one extraordinary individual, Dr. James McGaugh. As many of you know, Dr. McGaugh is a pioneer in neurobiology, renowned, of course, for his foundational work on memory. What you may not know is that when Dr. McGaugh arrived here at UC Irvine in the early 1960s, he didn't just join a department, he actually built one. In fact, he founded the very first department of neurobiology in the world, laying the groundwork for a revolution in brain science. But he didn't stop there. 2 decades later, he went on to establish the world's first center dedicated to the study of learning and memory. And that, of course, is the Center for the Neurobiology of Learning and Memory. His vision was clear: to create a place where brain research could thrive, where bold ideas and daring questions would have the freedom to grow and lead to groundbreaking discoveries. That vision, it's become a lasting legacy, one that sparked the creation of numerous departments at UC Irvine and centers at UC Irvine, and have inspired, of course, similar initiatives around the world. Dr. McGaugh has left an indelible mark on the field of neuroscience, and we're all beneficiaries of his vision. Today, the Center for the Neurobiology of Learning and Memory continues to thrive as a multidisciplinary center that's centered on 3 core pillars: advancing cutting-edge research, training the next generation of scientists, and engaging meaningfully with society. These pillars, they're not just academic goals. They really represent our commitment to making science both accessible and impactful. Our goal is to ensure that the knowledge that's created in the laboratories reaches the communities that can benefit most from it. And so now, it is my distinct pleasure to introduce the man who started it all. His vision, his dedication, and his leadership have created a legacy that continues to inspire neuroscience research and education across the globe. Please help me welcome Dr. James McGaugh to the stage to introduce tonight's speaker.
Wow. Well, thank you, Manuela, for that extravagant introduction. Very extravagant, I might say. But now it's my turn. It's my great pleasure to have the opportunity to introduce tonight's speaker, Dr. Michael Yassa, Professor of Neurobiology and Behavior and Director of the Center for the Neurobiology of Learning and Memory, and he also holds the James L. McGaugh Endowed Chair. He's one of us. Dr. Yassa did his graduate studies here at UCI. He received his PhD in our Department of Neurobiology and Behavior, and then had a brief career on the faculty of Johns Hopkins University before we very wisely recruited him back to UCI. His research is guided by questions of how memories are created, maintained, and sadly, lost. His extensive investigations are guided by insightful hypotheses, and his experiments employ creative use of investigative techniques and imaging technology. He has published an extensive and impressive number of scientific papers and his findings have been featured in many public media, television programs, as well as interviews and podcasts. He's not only an accomplished neuroscientist, he's also a superb teacher, and I know that firsthand because we taught a graduate course together. And importantly, he has received the UCI Faculty Mentoring Award. Dr. Yassa has received many other distinguished awards and honors, including the Ossoff Scholars Awards in Cognitive Disorders Research, the National Institute on Aging Distinguished Lectureship in Neuroscience, the Elliott Stiller Endowed Lectureship in Neuroscience Research, the Allen Edwards Endowed Lectureship Award, the UC Irvine Chancellor Fellow Award, and many other honors. We are delighted to have Dr. Michael Yassa here this evening to tell us about the beginning of the end for Alzheimer's disease. Please help me welcome one of our own, my colleague and friend, Professor Michael Yassa.
Thank you. Folks, give it up one more time for Dr. McGaugh. All right, I'm going to relocate over here. Right now, someone's world is disappearing. Not years from now, not months from now, but right now. A name they've known all their life, now gone. A face they've kissed goodnight countless times, now a stranger. A husband, a wife, a son, a daughter, erased. Imagine sitting across from someone you've known all your life, only to realize they no longer know who you are. Your name, forgotten. Your shared history, disappeared. Your presence met with concern, confusion, maybe fear. This is not simple forgetfulness. This is not normal aging. This is the slow erosion of a life. This is Alzheimer's disease. And in the last 60 seconds, 20 new cases were diagnosed. That's 20 families who have just begun to grieve someone who is still alive. Now, around the world, Alzheimer's disease is diagnosed every 3 seconds. The numbers are staggering. By 2050, 153 million cases. The global economic burden, even more staggering, $17 trillion. Folks, make no mistake about it, Alzheimer's disease is an existential threat to our humanity, and we must do everything in our power to end it. Now today, I'm going to talk to you about some of the things we know about the disease, some of the things we don't know. I'll also share with you a story about how our field might have gone astray a little bit, and we'll bring it back and talk about what we can do to move forward. And this is not going to be doom and gloom, by the way, just want you to know. I'm a messenger of hope here, not despair. So let's start with the basics. The first thing you should know about Alzheimer's disease is that we're not all equally at risk. Different risk factors impact each of us in different ways. The number one risk factor for Alzheimer's disease is getting older. We can't do anything about that. Getting older is a risk factor for Alzheimer's. In fact, every 5 years after the age of 60, the risk doubles. The next factor we can't do anything about is genetics. There are certain known risk factors, chief among them, you might have heard about it before, ApoE4. Carrying this gene also increases risk. Now, it does not mean that anyone with an ApoE4 gene will develop Alzheimer's. It just increases risk. Now, those two things we can do very little about. But there is a whole host of factors that we can do something about, what we call modifiable factors. Lifestyle factors, for example. Diet, exercise, social contact, heart health, all of those things can have an impact on our risk profile for Alzheimer's disease. In addition, our circumstance, our environment, a history of dealing with adversity, neighborhood, healthcare access, all of those things also impact our risk for Alzheimer's disease. And we'll talk more about some of these factors that can be modified towards the end of the talk. But let's go back to how this was discovered initially. What is Alzheimer's disease? What are we truly talking about here as far as the brain is concerned? And the story goes back to the early 1900s, when the gentleman on the left, that is Dr. Alois Alzheimer, first encountered a woman, a 51-year-old woman from Frankfurt by the name of Auguste Dieter. Now, this woman presented with a memory syndrome, but it was more than just memory loss. It was also a disruption in thinking, in judgment, in personality, and importantly, it was progressive. It changed over time. It got worse and worse over time. In 1906, when she passed away, he was able to peer deep into the brain under the microscope after autopsy and identified the first evidence of Alzheimer's pathologies, later termed Alzheimer's pathologies after him. The first thing he discovered were what he called senile plaques. These are plaques, they're exactly what they sound like, they're clumps of a protein called amyloid, and that protein tends to aggregate outside cells and presumably wreaks havoc on the brain. We're going to talk a little bit more later about whether or not that holds water, by the way. The other pathology is neurofibrillary tangles. That's another protein that also clumps, but inside the cell, and causes the cell to collapse upon itself and go through a cell death process. Typically, it's a marker of cell loss or neurodegeneration. And if you look at the brain of a patient with Alzheimer's disease as a whole and compare it to a healthy brain, you see a drastic difference. A brain with Alzheimer's disease is about 2/3 of the size of a healthy brain. The spaces that you see in between that got deeper, those are the sulci that got deeper, and the whole size has shrunk. The cortical mantle, so to speak, has shrunk. Now, this happens because of cell loss, because of neurodegeneration. And I want to be very clear, this does not happen in a healthy aging brain. This actually is a revision of our prior knowledge. We used to think that as we got older, we lost brain cells. Turns out that's not the case at all. We maintain our cells as we get older, but only in the context of a neurodegenerative illness like Alzheimer's disease are those cells lost. So this is unique. This is not just a consequence of aging. But that's the surface. Once you go inside the brain, there's a lot more going on. Inside the brain, you all have the same thing inside your brains, about 100 billion cells connecting to each other in about a quadrillion synapses. Imagine the information processing capacity that we have. It's mind-boggling. But all these cells require doing 3 things to stay healthy. They have to be able to talk to each other, to communicate. That's what brain cells do. They're designed for that purpose. They have to be able to carry out metabolism, and they have to repair themselves whenever they get injured. And Alzheimer's disease disrupts all 3 of these core functions. Makes it difficult for brain cells to communicate, makes it difficult for them to metabolize, and makes it difficult for them to regenerate or repair themselves after injury. And for about 35 years, the dominant thinking in the field was the cause of all of this was amyloid. This is the amyloid cascade hypothesis that was first proposed in the early 1990s by Dennis Selkoe and John Hardy. It's pictured here on the left. Everybody understand it? You get that, right? Okay, I put it up there just for show, but I simplify it for you. Now, what you need to know about this is very, very simple. Amyloid is the culprit. The idea is that amyloid is the thing that starts the chain reaction, the thing that starts it all. And through this chain reaction, it gets down all the way to dementia. So if we can just intervene at the level of amyloid, maybe we can fix this problem. That was the dominant thinking for more than 3 decades. And it has led to a couple of interesting things happening. One is animal models that people started to develop for Alzheimer's disease, all focused on amyloid. It made sense. Such a simple and elegant idea. Why not? It meant that if you design clinical trials to intervene, you're going to target amyloid. So all of the drug development that happens focused also on busting amyloid plaques in the brain. And this would all be fine and good if the amyloid hypothesis holds true. But let's take a look at the impact of this hypothesis. If you look at the sheer investment of resources and effort into this idea, going back and looking at how much the NIH, National Institutes of Health, has invested in Alzheimer's research, you see a couple of interesting trends. One, over the last 10 years, it has increased. That is a good thing. More and more research effort is devoted to Alzheimer's disease. About $40 billion over the last 24, 25 years. Now, you might look at this number and say, wow, that's staggering. But this is only about a third of what we've spent on cancer. So it's nowhere near where it needs to be. Okay? It's also about 0.01% of the federal budget. It's not a lot. Okay? So we do need to spend more effort, we do need to spend more resources on Alzheimer's research, but the problem is what was spent disproportionately went to amyloid-focused studies. 70% of that were studies that were focused on amyloid, and 30% focused on everything else. And if you think that's staggering, $300 billion were spent by Big Pharma on amyloid-busting therapies. Again, this would all be fine and good if amyloid truly was the culprit, if the hypothesis held true. But there are problems with this hypothesis. 3 problems in particular that I want to share with you. First, amyloid does not correlate with memory or cognitive decline. That relationship between amyloid and memory, which is like thing 1 in Alzheimer's disease, is very weak at best. Amyloid also is not associated with conversion to dementia. In other words, you can have a brain packed full of amyloid and never develop dementia. It's not a one-to-one relationship at all. In fact, 30% of older adults with amyloid in their brains do not go on to develop dementia. If it was truly the culprit, that would not be the case. But the most disturbing is that anti-amyloid therapies, which have been around now for some time, in fact, some of them recently just got approved. So FDA approvals have happened for some of these drugs, contentious approvals, because these drugs have limited efficacy and pretty serious side effects, including things like brain swelling and in some cases death. So again, if amyloid was the culprit, none of this would be happening. So what's the problem? The idea seemed so simple, so straightforward. Amyloid starts the chain reaction. If you intervene there, you can fix it. But we may have been looking in the wrong place. Maybe amyloid is more a symptom than a cause. Maybe it's just a small part of a much larger puzzle. And you'll get to see today that Alzheimer's is complicated. And some of that complexity comes from the fact that there are many contributors. Let's talk about some of those contributors. So some of the other candidates, for example, we talked about the connections and how important the connections are. We know that in Alzheimer's disease, there's a disruption in those connections, the synapses. And that tends to be much more closely tied to memory loss. We also know that there are changes in the brain's immune system. It wreaks havoc on the brain. It overactivates neuroinflammation in the brain, and that also has some severe consequences. There's also changes to the vasculature. As we get older, you'll hear me say this a lot, heart health is important for brain health. Well, one of the reasons is because the heart pumps all this blood to the brain to feed it and keep it well nourished. If there's any problems with the vascular supply to the brain feels that. That causes a condition that can lead to the path to the cognitive decline. And then finally, the last candidate that I want to talk to you about today is excitation inhibition imbalance. That's a mouthful, so I want to spend a few moments talking about what this means. If you think about the brain in a dynamic way, it truly is this beautiful, orchestrated symphony of activity. Electrical signals, chemical signals, flowing all throughout these brain cells. And it's a beautiful harmony, always kept in balance. That balance exists between excitatory signals, or go signals, and inhibitory signals, or stop signals. The brain is filled with stop and go and stop and go and stop and go, kept in beautiful harmony. It's like a yin and yang. But what happens when you disrupt this balance? Let's talk about the extremes. Let's say you have too much inhibition at the expense of excitation. Okay? Too little activity. That can lead to sedation. That's what you might see in something like coma, for example. The opposite situation, where you have too much excitation and too little inhibition, or too much activity, can lead to something like seizures. That makes sense. But a very interesting situation arises when you have just a little bit too much. Not enough to be seizures, not enough to be epilepsy, but just a little bit too much of a good thing, that now it turns out to be a bad thing. And to get this concept across, I'm going to rely on a 1970s Monty Python sketch. We're going to watch it together. Here we go.
Morning. Morning. What you got then?
Well, there's egg and bacon, egg, sausage and bacon, egg and spam, egg, bacon and spam, egg, bacon, sausage and spam, spam, bacon, sausage and spam, spam, egg, spam, spam, bacon and spam, 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 egg and spam, 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 baked beans, spam, spam, spam and spam. Oh, lobster thermidor crevettes with Mornay sauce, garnished with truffle pate brandy and a fried egg on top and spam. Have you got anything without spam in it? Well, a spam, egg, sausage and spam. There's not got much spam in it. I don't want any spam.
Why can't you have egg, bacon, spam and sausage?
That's got spam in it.
Not as much as spam, egg, sausage and spam.
Look, could I have egg, bacon, spam and sausage without the spam? What do you mean? I don't like spam.
Spam, 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 spam,
Sausage without the spam! Why not? It wouldn't be egg, bacon, spam and sausage, would it? I don't like spam!
Don't make a fuss, dear. I'll have your spam. I love it. I'm having spam, 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 baked beans, spam, spam and spam.
Baked beans are off!
Well, can I have spam instead?
Okay. How many of you have seen this before, by the way? Okay, some Monty Python lovers in the audience here. So the spam sketch, some of you might recognize, actually is the origin of how we use the word today. We don't typically refer to it just to refer to spam. We use it to refer to this, which all of us receive. All of us get spam in our email boxes and our messenger. In fact, you probably got a couple of spam messages about this very lecture. And the key with spam is that typically it's a good thing. It's a good thing to have communication, but too much of a good thing ends up being a bad thing. Too much noise drowns out signal. So the concept that I want to introduce to you today, you heard it here first, is brain spam. No, it's not this tasty little treat. We're talking about too much of a good thing in the brain. Too much activity, not to the level of epilepsy, but enough to disrupt the harmony, enough to disrupt the balance. And that can be an early sign of memory loss. Where do we see this first? In this little structure in the brain, the hippocampus. Now, you probably have heard about the hippocampus before. You heard that it's important for memory, and that is true. It is important for memory. But we're going to talk about what kind of memory today. We call it the hippocampus, by the way, because of its remarkable resemblance to a seahorse. At least in this crooked view, you can kind of see it. Okay, so what happens in the hippocampus, and how do we measure this? The first experimental evidence for this comes from animal models. This is from some of my colleagues back at Johns Hopkins, who stuck electrodes directly into the hippocampus of older rats. In particular, they had rats who had memory loss or memory deficits, and rats who didn't have memory loss or memory deficits. Because just like humans, there's individual differences. Some rats develop memory loss, some rats don't. I know, crazy, right? But if you're recording using these electrodes from the hippocampus of memory-impaired animals and memory-intact or unimpaired animals, you see a very interesting phenomenon. The animals who have memory impairments have a higher level of activity in the hippocampus. SPAM, too much of a good thing. A form of overdrive that imbalances this system. Years later, we did the same thing. We put folks in the MRI scanner, measured activity from their hippocampus, not with electrodes, but just using MRI. We're not going to go through the details of how this works. If you're interested, I'm more than happy to tell you about it. But we can measure the level of activity in the hippocampus using this technique. And we can do this in older adults who are intact, who are controls, who don't have memory loss, and look at it also in older adults who have mild cognitive impairment, or MCI. Thought of typically as an early stage of Alzheimer's disease, and we find the same exact thing. Patients with mild cognitive impairments also showed this overdrive, this heightened level of activity in the hippocampus. Okay, well, there's overdrive. That's all good and well. How do we know this is bad? How do we know this impacts memory in a negative way? Now, my lab has spent a long time trying to figure out the mechanisms for this and associating it with a number of different things. And I'll just give you kind of the summary image here. Hippocampal overdrive is bad for the brain. We know that it's not a healthy compensation. It's actually a dysfunctional condition. We know, for example, that hippocampal overdrive disrupts information processing, biases brains away from learning new things and much more towards remembering old things that we know. You may have heard before, can't teach an old dog new tricks. That's where that comes from. Learning new things becomes increasingly difficult because of this disruption in information processing balance. There's also a metabolic demand that is increased. That makes it also very difficult for the brain to maintain its energetics, another form of disruption. Another thing that we know the overdrive does is destroy cellular microstructure. It can actually, in and of itself, lead to cell death. And then finally, interestingly, there seems to be a bidirectional relationship with amyloid. So wherever you find amyloid protein, there's typically also overdrive. And wherever there's overdrive, amyloid protein tends to aggregate. Now, I'm not saying amyloid is the culprit here, but it may be related to this story in some subtle way. So all of this spells out bad news for the brain. So the idea is disruption of this balance, this hippocampus overdrive, leads to memory loss. But there's memory loss and there's memory loss. How do we measure this? How do we do this in an objective way? And I want to share with you how we do it in the laboratory. So the question is, how do we study memory? In fact, instead of telling you how we study memory, maybe I'll just show you how we study memory. I'll do you one better. How about we do a live experiment with you guys? You want to help me with this? We'll collect data from you all? Okay. This is going to be very straightforward, very easy. I just want you to study the pictures I'll show you on the screen. They're going to come up on the screen one at a time, couple seconds each. All I want you to do is focus on the screen. Ready? Here we go. Okay, how did that feel? 5 pictures, folks. Come on, it's not that bad. Now, in the lab, if we were to do this, we would do like 100. But just 5 is not too bad. Agreed? Okay. Now, I'm going to test your memory. And the test is going to be very straightforward. I'm going to show you 2 pictures on the screen, side by side. And I want you to tell me, left or right, which one you saw before. And I want to hear your voices all together. Okay? All right, here we go. Yes. Excellent. How about this one? Yes. I love the stragglers. There's always a voice coming out of the end. How about this one? Yes. Okay, how about this one? Yes. Fantastic. You guys did such a good job. This is like, you can go home and say, I took Yassa's memory test, I got 100%. It says, clean bill of health, everything is A-okay. Pretty straightforward test, right? But what if we made it just a little bit more challenging? What if we got a little bit trickier with it? Again, same exercise, left or right. I want you to tell me which one you saw before. In the same breath, all together, okay? There's a variety of opinions on this one. Oh, now you're changing your mind. Okay, it is the one on the right. But come on. That's a much tougher test. When I showed you a zebra earlier, you had no idea I'd test you with 2 zebras? I mean, who does that? But it turns out that's the kind of memory the hippocampus is really good at. And don't worry if this was challenging for you. It's challenging for everyone. It just gets more and more challenging as we get older, and then far more challenging for patients with Alzheimer's disease. But this is designed to be a challenging test. So that's how we measure it in the lab, and we've been able to discover that this memory overdrive is associated with more errors on this test. Okay? So that's the general idea. You have a test of hippocampal function, very, very specific, very sensitive, and now you can see evidence that this overdrive may be contributing to it. But it brings up a really, really important question. Is this thing reversible? Can we somehow figure out how to reverse this overdrive or rebalance this system, bring it back into perfect harmony? I wouldn't be telling you this if that wasn't the case. Kind of foreshadowing here. So that's what we tried to do. We did a clinical trial, and in this case we used a low dose of an anti-epileptic that is pretty well known, pretty regularly prescribed, called levetiracetam. You don't need to worry about remembering that at all. It works with a number of different drugs in that same class. But the key was it was a very low dose, because remember, this is not epilepsy. So for a patient with seizure disorder, a doctor might prescribe 1,000 or 2,000 milligrams. This was 125 milligrams. An order of magnitude lower. And there's an interesting lesson here, because when we started to talk to our clinician colleagues about this, we almost got laughed out of the room. Nobody believed that this would ever be an efficacious dose for anything. Okay? But the key was this was overdrive. It was subtle. It wasn't seizures. So the question is, can we use this approach to reduce this overdrive? And here are the results. So this is the trial that we did. This was a small study, again in MRI, with patients with mild cognitive impairment and healthy controls. In this case, they're also taking that task, that ZEBRA's task. We do it a little bit differently in the lab, but very similar kind of logic. The data that I'm showing you here first is the difference between MCI patients and controls. I showed you something like that before. It's exactly the same thing again. This is an independent replication in a totally different sample, showing the overdrive in MCI patients. In this case, those MCI patients were on placebo. Here's what happens on drug. The overdrive goes down. If you compare MCI patients against controls in terms of their memory performance, they make a lot more errors on this test, the ZEBRA's test. And with the drugs, their errors also went down. So not only did this kind of intervention reduce the overdrive, but it also improved memory. Now, this was a pretty small study, 17 patients and 17 controls. It was done in a 2-week exposure to the drug, which is very, very brief. The question is, what if you do a longer study with many more people and extend this over, say, an 18-month period, and instead of looking at just memory improvement, you're now looking for a staving off of cognitive decline? Can you actually see that you have somehow modified the disease process to make it so that they're not declining? And that is the study that was just concluded last year and published the results last year. A 40% decline, or 40% restoration in cognitive decline, rescue of cognitive decline. Now that's a big whopping effect, but it was nuanced. This was only in ApoE4 non-carriers. So it didn't work for everybody. It worked for a subgroup of all the patients tested, not for everyone. Now, you might look at this and say, well, that sounds like a promising new therapy, at least if I don't have the ApoE4 gene. And you may be right. It may be a promising new therapy. But I want to encourage you to think about this also slightly differently. This may also be a different way to think about Alzheimer's disease. See, we used to think about Alzheimer's disease as this. And maybe we need to think about it more like this. If you look at the aggregate body of literature over the last 30 years, even with the amyloid cascade hypothesis being there, it leads us to one inevitable conclusion, which is that Alzheimer's is not one disease. It is many diseases. And if we can shift our frame of reference, shift the way that we think about it, now we can start to make progress. So if we take this approach of identifying subgroups within Alzheimer's disease, you can start to apply precision medicine. Precision medicine would require us to take into consideration at the individual level all of their individual risk factors, would require us to use something like biomarkers, which is something we do regularly in my laboratory. So it could be imaging, it could be sensor technology that you're using, it could be heart rate variability, retinal scans, digital cognitive testing, like this test that we showed you with the zebras. All of these things can give us indications of biological mechanisms that are happening in the brain, and we can link them all to the kinds of mechanisms that are changing in Alzheimer's disease. You take all of that information and you feed it into our favorite word these days, AI. There's a process of knowledge integration where you take all this information, synthesize it, and based on that, try to do drug discovery in a very different way than we used to do it. Instead of co-opting drugs that have been around for quite a while, now we're talking about targeting things based on the biology, based on the mechanisms. And you can design the right kinds of clinical trials to also be adaptive, to take into consideration biomarkers. This is the stuff that we're focused on now. And we're hoping that using this approach, one can design the right intervention for the right group of individuals. Now, you might look at this and say, well, that sounds a little bit futuristic, sounds a little out there, sounds a little fringe, but this should be familiar to anyone who's been looking at precision medicine for quite some time. This is the cancer playbook. This is familiar to us, folks. We've known about this for a long time. We know what works because we've applied precision medicine to cancer, but not yet to Alzheimer's disease. This requires a paradigm shift. Now, paradigm shifts, according to the person who coined that term, Thomas Kuhn, in his magnum opus, The Structure of Scientific Revolutions, he talked about these paradigm shifts. They're not piecemeal processes. They're actually quite painstaking. They require an adjustment of the constellation of beliefs, values, and techniques shared by a scientific community. Translation, it's hard and it takes a long time. And especially hard for Alzheimer's disease, it's not just one paradigm shift that we need. It's actually two paradigm shifts that we need to undergo. The first one is from amyloid to other targets. I hope I've convinced you amyloid is likely not the culprit. But also, it's a shift from a single cure model to a multiple intervention model. Different drugs, different interventions for different patient classes. The biological mechanisms for different groups may be very different and may require different combinations of interventions. Now again, this sounds like it's out there, but I told you I'm a messenger of hope today, and I want to share with you that this is one of the reasons I'm very hopeful. Just a few months ago, the National Institute on Aging held its Alzheimer's Summit, Research Summit. And the topic there was precision medicine applied to Alzheimer's research. It seems like the ideas are finally starting to take hold. Are we there yet? No. Are we closer? Yeah, I think so. The fact that this is the kind of conference that brings together all of the researchers around the country that are doing this work and is influenced by this idea brings me hope. So those are two paradigm shifts, but I want to share with you a third paradigm shift, one that I think you can start to apply today. So the third paradigm shift, which is really, really important, is the shift from cure to prevention. Now, there's a lot of things that I can put on the list of what can we do to protect our brains. It's a very, very long list. But I've selected 4 things that I want to share with you because they have the best evidence in their favor. And when I say best evidence, I don't just mean public health epidemiological evidence, I mean also clinical trials. Okay? So those 4 are selected because they have the best evidence to show that these things actually work. They actually reduce your risk for Alzheimer's disease. The first one, I don't know if you're going to like this. Every time I put this up, people go, oh, I know, it's physical activity, exercise. Uniformly, exercise seems to be associated with better health outcomes in general, but reducing risk for Alzheimer's disease with physical activity. Isn't that incredible? And before you ask me how much and what kind of exercise, I'll give you a couple of formulas. Usually sessions more than 30 minutes are better than sessions that are less than 30 minutes, duh. Works for aerobic exercise, works for strength training, all of it is good. We've even shown that walking as little as 10 minutes can improve memory retention. So it doesn't take much. So I would say, concerning quantity and quality and all of that, any exercise is better than no exercise. Okay? So beyond exercise, what else is there? Here's one maybe you haven't thought about too much. Social and cognitive stimulation. And I want to take a moment to talk about social. Social contact is one of those incredible things that we do as humans. It maintains our sanity. It allows us to relate to others. And it turns out it also protects our aging brains. And this is so important. As we get older, get to retirement age, sometimes that is associated with a fork in the road where you can either remain socially interconnected with folks or you may not. And those who go into social isolation are at an increased risk for Alzheimer's disease. So maintaining that social contact, that social connectivity is really important, and it also reduces risk for Alzheimer's disease. The third thing, diet. Again, we can go through the list of diets. I'm on this diet, I'm on that diet, which one works, which one doesn't. And I can tell you right now, all of the fad diets out there, nothing has worked as well as the Mediterranean diet. So the Mediterranean diet is pretty straightforward. It's leafy green vegetables, it's fruits and nuts. It's rich in the good fats, low in the bad fats. Protein mostly from fish sources, and low on red meat. So if you do enjoy a steak every now and then, it's perfectly fine. But again, being on the Mediterranean diet, and the clinical trials, not just epidemiological data here. So actually putting people on this diet seems to reduce their risk for Alzheimer's disease, and among those with Alzheimer's disease, it's associated with lower mortality. So in general, this is a pretty good sort of prescription for a diet. Then the last thing I want to share with you. Everyone does it, but every species does it, not just us. And it's somewhat a mystery why every species under the sun has to sleep. We spend about 1/3 of our lives sleeping. You'd think maybe that's important. Turns out it's really, really important. One, as we sleep at night, our brain is clearing through metabolic waste. So if you don't sleep enough, that's not happening as well. And two, studies have shown that if you have sleep disruption, you're at increased risk for Alzheimer's disease. So if there's sleep loss, quality, quantity, that can be a problem. Getting enough sleep will do its thing to reduce risk. And again, when I say enough sleep, I mean quality and quantity. Just being in bed at night does not count. Just being in bed and doom scrolling does not count. We're talking about actually sleeping. Okay, 4 things. You gonna remember those? Okay. Now, I hope that you're seeing that there's lots and lots of moving pieces to this puzzle. Lots of parts. And we don't have all the parts yet. We're still trying to piece it together. There's complexity to Alzheimer's disease. But what we've learned from the amyloid experiment is that we have to embrace the complexity. We can't run away from it. We can't just settle for simple solutions. Even if they're elegant, even if they're beautiful, and they seem to make sense. Complexity is reality, and it requires a lot of effort. It requires a lot of people, a lot of teams, to put together the pieces in this puzzle. Teams from all around the world. This is my team here at UCI. Some of them are here. Raise your hand. Raise your hand if you're part of the team. I see some of them up here. This is an incredible group of people that I am privileged to work alongside every day. And part of my privilege, of course, is seeing their passion and commitment and their leadership to be able to identify, discover solutions, treatments, interventions, mechanisms for Alzheimer's disease. We're bound together by this joint purpose. And it's just such a delight and such a joy to be able to work with them. But of course, we don't do this in isolation. We are also situated in this incredible center that Jim McGaugh built. So thank you, Jim. And for me to have the privilege for close to 10 years now being director of the center, it's really incredible. It has provided, aside from providing the fountain of knowledge and innovation and discovery that it does for everyone else, it has provided a home for my research program and my team, my crew, and the work that we're doing in a way that I could have never imagined was possible, even back at Johns Hopkins where I was there. So coming back to UCI was absolutely the right thing for me to do. I want to be buried here. I'll go on the record. I will say this. But it's been a wonderful place to see this kind of research through. So I want to leave you just with one note here. And that is that the end of Alzheimer's disease begins with the courage to think differently. And I hope that each of you will walk out of here today thinking just a little bit differently about Alzheimer's disease, about what we need to do, about the effort that it takes. And if we can all do that, then maybe we can start to see the light at the end of the tunnel. Thank you.